Jessica.
Okay, let's see if I can get, there we go. All right, I'm very excited to share, some relatively recently published work on how amino acids can impact important inorganic structures for prebiotic environments. So the structures we're going to be talking about are chemical garden structures, and these are very well studied in a number of different fields. But the gist of it is that these chemical gardens occur when you add an inorganic precipitate, like a metal ion, in our case iron, to a solution with ionic materials. And you can get a variety of different structures for these. Like I said, they can be used in the field of physics, material science, chemistry. They're really well studied and very diverse. In our case, we wanted to use them as analogs for hydrothermal vent settings. This is one hypothesis of where the origin of life could have occurred. And it's important not just on Earth, but on other worlds like early Mars or ocean worlds such as Enceladus. So I think that these are really, really simple structures that we can use to just get a better idea of how these large, complex structures behave. There has been a bit of work done looking at additives to these chemical gardens, specifically phosphorus, which is also very relevant for origin of life. But we really didn't find any examples of how organics can impact the structures, the growth of chemical gardens. And as we all know, as you saw in the previous talks, organics are very important for the origin of life. And specifically, this talk is going to focus on amino acids, which, as Jay stated, are very important for the formation of peptides and proteins. So we wanted to take amino acids and test the effect of organics on these chemical garden precipitates. In order to explore this, we chose 5 amino acids. We wanted to really vary the functional group on the amino acids. So all amino acids have an amine and a carboxylic acid, but we wanted to make sure the side chain was different between the amino acids. So we chose glycine and alanine. These are the simplest amino acids, and they're very often used in prebiotic chemistry and origin of life research. Although we wanted to, again, vary it to make sure we had some interesting opportunities for reactivity. So we included cysteine, which has a thiol group, which is known to be redox active, as well as lysine and aspartic acid that contain an amine and a carboxylic acid, but at very different carbon chain lengths. So we thought that this was a nice mixture of amino acids that we could really test the effect of the size of the amino acid as well as the functional groups present. In order to prepare the chemical gardens, we created a solution of water and iron silicate, or water and silicate, and then added our iron. We dissolved the amino acids in the water prior to addition of the iron, and we tested a number of concentrations ranging from 0, our controls, to 200 millimolar concentration. So here's just an example of the chemical garden's growth. This was grown with the presence of 20 millimolar glycine. And so you can see the growth starts very fast. These are very exciting experiments to run. Immediately you start to see some growth, and then by 5 minutes, you have a really complex network of morphologies, which is very nice. I won't belabor the point too much, but these grow very quickly, and we observe different morphologies, in each of the reactions. Here's an example ranging from 5 millimolar glycine to 200 millimolar glycine. And so across the board, we didn't see any obvious differences between the amino acids used. I mean, we grew these in triplicate, and there is variation from growth to growth, but they were all, you can see they're all, roughly the same height. We see similar structures. The higher concentrations tended to be slightly shorter, but even that was, we really couldn't tell exactly what was going on there. So in order to get a better idea without just relying on, what we were visually seeing with the naked eye, we wanted to analyze our chemical gardens with microscopy. So we were able to mount a series for SEM and get a variety of morphologies. But a really important part about this is that the workup of the gardens played a large role in what we were able to get. And so we found that we really needed to make sure that these were grown in plastic tubes as opposed to glass and filtered very carefully. They're very fragile structures, and we tested a number of filter papers. I won't, I know we're short on time, so I won't go into this detail, but I would be happy to discuss the sample prep of delicate materials with anyone at a later date. But it really was very important because this is what the chemical gardens looked like when we were able to get good work up, and this is what they were able to look like when we did not work them up properly. So there was a very, very clear distinction in our ability to get good data that I just wanted to briefly emphasize. And so we were able to get some really excellent images of our chemical gardens via the SEM. Here's just some examples of our controls. You'll notice we see a number of different morphologies that we were able to access here. But notably, and this will become important in the coming slides, the structures are very rough. So you can very clearly see that the edges and inside are very rough for this material. And then here's an example of alanine, a bulb of alanine grown from, you know, something like this. And we noticed that the edges of this were much smoother at 5 millimolar. And here's a direct comparison. So you can see in the control, it's very rough. It is uniform throughout. However, when you start to look at the 5 millimolar of amino acid, in this case alanine, we see a small layer of smooth material on top of this rough structure. And this is really only further emphasized as you go up in concentration. So this is comparing 5 and 20 millimolar alanine. So you can see there's a thin layer here. However, when you, excuse me, not 20, 200. When you get up to 200, we see significant layering of this smooth layer. And so we believe that, you know, there's nothing else in these reactions except for the amino acid, the ion, the water, and then the iron. So we suspected that the amino acid was interacting with the chemical gardens to form this layer. And so that leaves the question, what is happening in solution? So we briefly took NMRs of all of our solutions, really to just test to see if there was any reactivity, any kind of, you know, polymer formation or any decomposition. And NMR is a really common tool that's really great for analyzing, you know, the protons of these such spectra. And we really only saw what we put in. So for this NMR, you can see we have a nice water peak and then the glycine peak, and that's it. And so this indicates that we're not seeing any significant reactivity in our solution. We do see a slight broadening of peaks, which is typical for materials that have iron in it. Iron is also magnetic, which leads to broadening. So no organic reactivity here. However, we really wanted to confirm that the layer, the smooth layer, was from the organics we added. So looking at the EDS data, which will tell you the elemental composition, a little bit about the elemental composition of these layers, you can see that we looked at the inner layer and the outer layer. And, you know, for both we see iron, for both we see silica. However, in the outer layer, we see sulfur. And this is a chemical garden grown with 20 millimolar of cysteine. So the only place you're going to get sulfur from is the cysteine amino acid. And so from this, we concluded that since the sulfur is visible on the outer layer of the surface and not the rough internal layer, the amino acid must be on the outer smooth layer. And that's what the smooth layer corresponds to. And so specifically in regards to prebiotic chemistry, you know, these chemical gardens are very uniform in shape and tend to look very biological in nature if you're looking at them. And especially even if we go to another world, we see this very uniform biological shape and we detect organics. This could lead to a lot of false positives for origin of life. So we want to make sure, you know, when we're looking at this, that morphology is not the only thing we're looking at for life detection. Additionally, these organics and inorganics can interact in complex ways, which leads to further questions, where it's how does the binding and concentration of these organics impact the reactivity? You know, at some point, concentration of organics is good to get reactions. However, you know, you want to be able to see the metal and interact at that center. And then could possibly the concentration increase reactivity at these sites? And so my conclusions are that the presence of organics clearly impacted the growth and structure of these chemical gardens. The concentration of the amino acids had a higher impact than the species of the amino acids, so we didn't see much of an effect of the different side chains. No organic reactions, specifically polymerization, were observed, and the organic is localized and on the outside of the chemical garden tubes and appears as a smooth layer along an otherwise rough surface. I think I am running low on time, so I'll just quickly thank everyone. I'd like to thank my postdoc advisor, Lori Barge, who's been really excellent to work with. I've been able to, propose a lot of new ideas and bring, different expertise in the lab, which is great. Scott Pearl is the other co-I of the lab and has also been really fantastic to work with. Michelle Hooks is the graduate student who performed most of this work, and I really enjoyed mentoring her. Paul Webster helped us a lot with the SEM and was very, very helpful for sample prep. I'd like to thank Icy Worlds for the funding, the rest of my lab for being a pleasure to work with, all of you for your kind attention, and I'd be happy to take any questions.
Good job. Thanks, Jessica. Everybody staying on time. That's great. So we got about 3 or 4 minutes here if there are other questions for Jessica. Maybe a little brief discussion of prep work, which I imagine was quite laborious.
Yeah. We found that to be challenging. Absolutely very challenging.
I am curious, Jessica, since I don't see any other questions here in the box. The idea would be, obviously, we would need to be somewhere in some sort of semi-oceanic or estuarine type of system, right? So this would be an icy worlds possibly thing there. So yeah, and you suggested there could be some concern about us finding those things and then sort of false positively identifying them as perhaps living structures. Is that a real concern for these things?
Well,
it's kind of a forensics.
I think that when looking for biosignatures, and we really have this one example for life, right? Like it's just us, right? So I think when we're looking for life on other worlds, we just need to be really careful because you can very easily imagine someone detecting organics, detecting amino acids even in this very regimented structure that may look biological. And I think that this is just a cautionary tale, for that. I think that there's a lot of people doing really great biosignature work that is far out of the scope of my talk, but it's just one thing that we clearly noticed. These are very uniform structures, and especially with the organics added on, I think it could look very biological.
And then, if I can just add quickly to that, so ultimately the goal would be then to produce these things and then to presumably look at how sort of species would undergo reactivity within, on, throughout them, which I know has been some of the focus of your work.
I know. This is, of course, we did set ourselves up a little bit for no reactivity. Amino acids are. We didn't suspect we were going to get a lot of reactivity here, but we do want to go back and test, more reactive organics, simpler organics, to really see what we can get.
Right. Okay.
For that.
Yeah. Awesome. Okay. Awesome talk. You guys are doing awesome work always out there. Any other last minute questions? We got about a minute or so before we transition here to Ben. I don't see anything in the box, so you get the prize, Jessica, for finishing early. Awesome. Thank you so much.